I'm at Bach Tower with my wife, partner, and we're going to be walking around with Cheryl Peterson. In fact, I'm shooting B-roll right now in my introduction. I've already done the walkthrough with Cheryl, so I like to do that because then I know what happened and I can sort of, rather than just guessing what you're going to experience, we go through, there's a greenhouse facility down there, there are bed gardens down here. We're going to look at some species in Lamiaceae, we're going to, which are the Conrandina and the Dicerandra, and also the native jujube. There's a lot of work being done there, as well as a few other things. There's the Crotalaria avenensis, the, I've already forgotten the genus, but it's the Hillsborough aster. And so there's a bunch of stuff going on over there. And so let's get going. I'll string this together in sequence, and hopefully you guys will enjoy this. It'll be in multiple chapters too.
Flowered, and they're dying back while the new growth is coming up. Go ahead and take a leaf of that and smell it and eat it. This one I'd recommend eating because it's chemically related to peppermint. It makes a great tea.
Yeah, I can, despite my lack of, my inability to smell, I can get that.
This is one that no longer exists in wild populations because the one population was found on a new development area, all private property, and so its population was destroyed in favor of a house. Because that's just what happens.
Yeah.
We have been working with a local state park next to where the natural population was found in South St. Lucie County.
Okay.
To introduce populations onto the managed lands there.
Okay.
And those populations they're doing okay, they're hanging on, but it needs management, and we really have to work with the park to make sure that it's not going to go extinct there. Because basically the few plants here is all there is left.
Wow, and what's
There's another couple beds of them, but yeah, because they only live about 1.5 years-ish on average.
Okay.
And so a lot of the plants that were in here have already died back, some are dying, and so we're going to need to take cuttings to repropagate those clones.
Okay.
When we first got involved in this plant, there was like 89 plants left in the wild. So we cloned all of those and keep them redundantly cloned, and that material gets used to take cuttings to introduce into the wild.
Okay.
And the wild population, or the populations that we've introduced, had as many as 4,000 to 5,000 plants, but as management doesn't happen as much, or there's drought or something like that, the populations have declined, and we're trying to build them back up.
Okay, okay. And
drought is really getting to a lot. The spring drought is really getting to a lot of the species.
Okay.
Because they produce seedlings about this time, and they require, like once the seeds drop in, like say, December, January, they need the winter rains. We don't get a lot of winter rains, but we get enough to imbibe the seeds, and we need enough regular rainfall just to be able to keep the seeds alive. And the strategy of these plants, of a lot of our upland ridge plants, not just the Lake Wales Ridge, but even these from the East Coast, the little seedlings, as the seed germinates, it'll form like these really long, thin, hair-like roots that go down and try to find any moisture. And a few feet below, they might find some moisture, and then they're like, okay, I can live here. And then they use that to be able to form the cotyledons and true leaves.
Okay.
And so that's what they do in the wild. But if they're just starting to form those hair-like roots, or if the moisture level goes down even further, they'll die. The one natural population of this one year, in maybe February, March, there was like maybe 8,000 seedlings that I saw. A month later, because of drought, maybe a month and a half later, they were all dead.
Wow.
Yeah,
wow.
and it's only because of the drought.
Jeez.
Yeah, and the seeds do not persist in the soil, so it needs seedlings every year, and that's really a problem with some of these species.
Yeah, it sounds like it. What's the binomial on this?
This is Savannah's mint. It's Dicerandra maculata variety Savanera.
Savanera. Okay, I had asked you about that. Last time when I saw you at Hallstrom, we were talking about it a little bit. So good to meet it.
Yeah. It looks very much like the Lacula's mint, and except it's got like the wider leaves.
A little broader,
it's a little bit more robust, a little more cumulative. Just upright and stronger, so it's just a little bit more robust. I wondered if it was maybe a tetraploid and the other one was diploid, so we did like ploidy studies. Everything's diploid, any population,
Okay.
they're all the same, so it's just something inherent in this.
Okay.
Yeah.
Is there an effort to split it out of Immaculata, or are you going
I think it should be.
Yeah, you
I
were
have to
saying
treat
that.
it as separate, yeah.
Okay.
And because this was thought to be a subspecies of Lacculus mint, because there was more of that, and they both have spotless flowers, which is why they're Immaculata.
Okay.
But when the University of Florida, the Soltis lab, did some genetics on it, it looks, from what I remember, it looked like this was ancestral to the Lacculus mint. So maybe that,
It's
that should
radiating
be a
away.
subset of these. But to me, I'd love to see them separate, so they can be treated as separately and both be listed separately.
Okay. Well, hopefully it can meet that criteria to be properly split out.
Yeah, I don't want to step into the world of taxonomy.
Once again, yeah, because that's a hornet's nest. Yeah. Oh, it's so great to meet it, though.
Yeah.
I was at Savannah's not too long ago, and I just kept an eye out. I knew I would probably not see it, but I was just hopeful that I might cross it at Savannah's. But
There's a
good
couple places that are probably accessible if you know where to go, but other than that, you need to get in a truck and drive up the railroad track, and then know where to stop in the woods.
Oh, okay,
So it's
okay.
kinda
Which is good.
Yeah.
Which is good. Yeah.
That's nice. Preserved.
Okay.
There's even some at Hallstrom, but
Okay.
not as much. We've seen it there. This is not a rare plant.
Okay.
It is a Central Florida plant, the Lupinus diffusus.
Okay.
The rare one looks very, very similar, except it's got pink flowers. And if you pull one of these open, you see these stipules in here. The rare one does not have stipules.
Okay.
So if you see a species like this that has pink flowers and a lack of stipules, you know.
You're into something special.
Yeah, exactly. If that would be the Lupinus aridorum, the scrub lupin.
Okay.
There is another pink one with bigger leaves, but that's further north.
Okay.
Anywhere in Central Florida. Not really anywhere. This one can grow a lot of different locations, roadside, sandhill, some of the scrubs. You see it all over.
Okay.
But this one, the rare one rather, can only grow on the lake, the Winter Haven Ridge in a couple of areas.
Okay.
And the Mount Dora Ridge, which is around the SeaWorld area. And we've done a bunch of rescues up there because of development, I-4 widening, etc. We have one more rescue up there.
Okay.
Basically, it's almost gone up there. There's one spot that we're going to try to help restore. But basically, it has to grow, the rare one has to grow in that soil up there and on the Winter Haven Ridge. It cannot grow on the Lake Wales Ridge.
Okay.
Even on white sand, the right soil type, it cannot.
Okay.
But so we did some studies with chemists and with looking at the soil. We did some genetic work on the nodules that it formed. Being in the Fabaceae, it forms the symbiont, the
Right,
nodules,
the
on the
nitrogenizing,
roots. The nitrogen fixers. And so we did DNA extractions of what's in the nodules to try to find what species are in there. We just looked at the bacteria because that's all we had funding for.
Okay.
But what we found was that there's species that are found in the common one, different locations, that are like a wide range of just common nitrogen fixers.
Okay.
So it kind of doesn't care what it talks to, it's like, anybody fix nitrogen for me, you can have a home. Where the rare one has specialized to need a specific species or two that's only found in a couple areas.
Okay.
And we're trying to figure out why. So we worked with a chemist from Florida Institute of Technology.
Oh, okay.
And he was looking at all the chemical exudates that came out of the roots. And because apparently what these species do is they form the root and then they send out chemical signals that get heard and answered by microbes that are in the soil, and so they speak to each other that way.
Okay.
And so it's almost like a energy saving strategy. If you're a plant, you only have so much energy.
Sure.
Are you going to put it into creating all these root chemicals, exudates that will talk to anybody, and you put tons of energy out there and hoping that somebody will answer? Or do you just go, I know where I grow, I know what's here, I'm going to choose one and talk to it. You know, and so this one chose one strategy, the other one seemed to choose the other strategy, which I find really interesting because they're very, very similar genetically, except for certain things like that. But because of development and everything else, that's why this one is not listed, and the other one is so rare and pretty much almost gone from Orange County because of development, because it can't grow anywhere else except there. So, yeah, so that was just really interesting that we learned what its strategies are and that it needs specific soil symbionts.
Yeah, and that goes back again to what we were saying a minute ago off camera with the hologenome and the holobionts, I should say, and niche construction. So niche construction in the sense that you, like you said, one is specialized, one is more generalized, and you know that's interesting, and that loops back to selection and fitness and all these things. But I love that breakout that you have to think a little more dynamically about the relationship and the relata and all of that.
And
so that's fascinating.
In order to do population introductions of the rare one, we could take soil samples and do DNA extractions, and and look to see if we can find the required symbiont in the introduction site. And also, as the site gets managed over time, we could see if that helps, if that's going to help the soil symbiont component for the rare species. So there's applications to that that we would like to explore.
Oh, I love that. That's really neat.